Yeah, so thank you, Alper, Artur, and Onur for letting me talk today. I'm going to be talking about the analysis primarily of single-cell RNA-seq data, but I will give some background so that if you guys encounter single-cell data or if you end up doing your own experiment and you generate some single-cell data, that it won't really be a black box as far as what's going on and what the goals are when you're actually working with this data. So a little overview, I'm going to talk about, just to start with, the two data sets that we will work with today that Artur has put on the Biocore website. We'll go over the links in a minute. Then I'm going to talk a little bit about why you would actually use single cell, focus on the in-drops technology that the data we're going to work with today was generated with. There are many other single cell methods out there. This is just one of them that our lab happens to have on site. A little bit about DolphinX pipelines, which are used to process the raw sequencing files from the sequencer all the way to what we're going to work with today, which is a UMI matrix, and we'll go over that in more detail at the very end as well when we talk about the data structures. So first, a little bit about the data set that we're working with. We're going to work with two separate data sets. Essentially, I'll show you some commands and some processing steps that you would use regardless of the package that you're going to use for analyzing your single cell data. And first, we'll apply it to this BMDC data set that we have. So for those who aren't familiar, what you can do is take bone marrow from mice, and you get the progenitor stem cells. If you culture this bone marrow with GM-CSF, which is just a cytokine or a chemokine, for about 9 days, what you get out are immature dendritic cells. You can then incubate these dendritic cells with LPS, and they have a large transcriptional response to this stimulation. So you take the immature DCs, you add LPS, and you get mature dendritic cells that are responding to the LPS. So this is the first data set that we'll work with today, and I'll kind of walk you through how to analyze it. The second data set that we'll work with is a subset of a data set that our lab has been collecting for a couple of years at this point, where in collaboration with the Harris lab on campus, we in the top use suction blistering, so they recruit vitiligo patients. If you notice in one of these panels, the patient has white blotchy spots on their skin. This is where melanocytes are being targeted in an autoimmune reaction, and they are dying. So you get this lack of pigment. So we have these suction blisters where we form these blisters and can extract out the supernatant, which contains the cells that are there. And then we run them through the single cell method, which again, in this case is in-drop, but we've also done 10x, we have done drop-seq. So if you're curious about one of those particular methods, feel free to ask. So again, we'll start with the BMDC data. I'll show you how everything kind of works. And then you guys will have the freedom to apply these same techniques to this skin data. And one of the biggest things that can be I don't want to call it a learning curve, but kind of having the confidence that to take in a single cell data and to analyze it and process it and annotate it with all the cell types that you think are there, there's not like there's not 100% right or wrong way to do this. And in some way, it is a little bit subjective. So when you're working with the skin data set, feel free to try different parameters, do things other than what are exactly what's written here. Because in the end, you're going to be the one who tells us what's going on with this data. And there's not necessarily a right or wrong answer, but there are some answers that are definitely closer to what the biology would be than others. So why do people even do single cell? The idea is that for the longest time, you would take a solid diverse tissue, so liver, tumor, whatever the case may be. You would have to do minimal prep, so essentially just an RNA isolation. And then you would do bulk RNA sequencing. And what you get from this experiment is a single gene expression value for every gene that was expressed in the data. However, if we compare that to single cell, what you're going to do is take that solid diverse tissue and you're going to dissociate it into a single cell suspension. So now when you run your single cell experiment, instead of getting just a single value, so say actin beta is expressed at 10 TPMs, instead of only getting that single value like you would with a bulk experiment, you actually get a distribution of values where in the bottom right here, when we look at the distribution of expression of this gene, the actual gene doesn't matter. For every single cell, we can track exactly how much of that transcript was present in that single cell. And so if we have, say, 1,000 single cells in our experiment, you might have a distribution of gene expressions instead of just a single value. Now, the key part here is that for single cell, if you have a heterogeneous population where there's either multiple cell types, there might be multiple cell states, oftentimes between these different groups, cell type A or cell type B, you're going to have a completely different distribution of gene expressions for the gene that you're looking at. So some genes might not be different at all between these red and green cells. So, just like a qPCR experiment where you might use a ribosomal protein or GAPDH for normalizing your qPCR data, the same principle holds here. There are some genes that are going to be the same regardless of which cell type that you're looking at. But at the same time, there are some genes that are only expressed in a single cell type. And so those are genes where you're going to have two discrete populations as far as the distribution of gene expression values. So not all problems necessitate a single cell RNA-seq. Many problems are still perfectly addressable with bulk RNA-seq, and you don't need to go the extra step to do single cell. But there are cases when it does become very powerful, particularly when you have heterogeneous populations and you expect there might be different distributions of gene expression between these different groups. It's very powerful for studying intra and intercellular variations in gene expression. And the last thing that we'll kind of use it for primarily today is that it's very useful for unbiased discovery. If you think about a flow cytometry experiment where you take a sample and you stain it with antibodies and then you bring it to the flow core and you can read out how much each cell is expressing or at least stained for those proteins that you tag them with. The problem, not problem, but one of the limitations of that approach is that you have to know what you're looking for to start with. You have to know exactly what protein you want to stand for, whether or not those proteins are good markers for the cell type that you're looking at. If you contrast that with single cell, in single cell, you don't have to have any prior notions about what might be in your data. You don't have to expect a T cell. You don't have to expect a macrophage and design a panel based on those expectations. Instead, you can just in an unbiased way take your whole single cell suspension, run it through single cell, and you will read out the transcriptome of all of those cells and be able to use that to build up a map of whatever sample it is that you're working with. So single flow cell workflows, again, there are many different technologies out there right now. If you have a particular question about a particular technique that you might have come across, feel free to ask, and I may or may not be able to give insight depending on if I've used that method before. But they all follow a core set of principles where you're going to take your solid tissue, I guess it could be cell culture to start with, and you're going to dissociate that into a single cell suspension. Alper and Onur, can you guys hear me? I hear Slack going off. Do you guys just mind grabbing my attention if it's something that you want me to stop and go over?
Yeah, don't worry.
Okay, yeah, just because I can't see those, see that right now.
Continue, no worries.
Okay, cool. So you're going to take your solid tissue, you're going to dissociate it into a single cell isolation. And then depending on the method, some methods are droplet based, where you're going to use microfluidics and you're going to take each one of these single cells and put it in a small droplet, kind of in an emulsion. Other techniques, you'll flow sort them and put them into a 96 well plate. So it's the actual plastics of the plate that are responsible for isolating each single cell. But either way, you end with a single cell and a single contained kind of reaction unit. And you're going to do the RT, which is taking the cell's RNA and reverse transcribing it into cDNA. From here, the end goal is that you want to amplify your library to have enough material to sequence. There are different ways to do this. Some methods will use IVT, so they'll take that cDNA that you've just created and make many, many copies through IVT, which will make RNA. Then you can take that RNA, RT it again, and get it ready for sequencing. Other methods will take that cDNA and just use PCR to amplify out all of the transcripts that you want. The reason that there's kind of these two different approaches here is that people found that PCR does have some biases. So depending on the primers that you use, the annealing temperature, GC content, you might be more or less likely to preferentially select out and amplify transcripts just based on those parameters. IVT, because it's not an exponential, it's a linear amplification, and they found that it just had less biases when you're actually going to amplify out the library. But either way, after IVT or PCR, you'll have enough material for your library, and you can put it on the sequencer. What we're going to focus on today is really what's in the blue square, which is that we have sequencing data, and we're going to build up the single cell expression profiles and work to identify what cell types are actually in that data. If you do have questions about some of the previous steps, again, feel free to just reach out with or ask at any point. So today, the data that we are going to look at was generated with the InDrops technology. So I had mentioned that some methods are using microfluidics. Other methods use, a 96 well or a 384 well plate. InDrops is one of the microfluidics based techniques. 10x, which is probably at this point the most familiar name as far as single cell. If you read it in a paper, most of the time it's going to be 10x. They are also an emulsion-based single cell method. So everything that we're looking at here is going to be just as valid if you encounter 10x data. The only difference is going to be the chemistries that they're employing in order to generate the actual libraries, but the physical principles of it are the same. So this diagram goes right to left, actually. And you can see that there's all these little channels and cells are coming through from the right. Again, going to the left, you can see a little red arrow pointing at a cell. That cell flows through this device and it mixes with another channel that has RT reagents and lysis reagents, as well as the beads that enable this to actually work. And I'll go over what those beads exactly are. All three of those things, the RT, the cells, the beads, combine together and then get pinched off from some oil streams. And they form these little tiny droplets that you see all the way to the left. These droplets, there's chemicals in here that make it so they don't merge together, they don't coalesce, they remain discrete emulsions. So if you've done or heard of emulsion PCR, which is a pretty old technique, but it obeys the same principle. You have these small isolated droplets, and in every one of these droplets, you have a RT reaction going on, and it's isolated from every other droplet. So when you take each one of these droplets, every one has its own single cell in it. And in parallel, you're doing this RT reaction in thousands or tens of thousands of cells at a time. Going into a tiny bit more detail of what's going on here, the lysis and RT mix, you form the encapsulation in droplets. And on these beads, these hydrogel beads, so in InDrop, they're hydrogel beads. In 10x, it's also hydrogel beads. They have these beads loaded with oligos. And when you release the primer, you're starting the RT reaction within the droplet. Now, the question really is, what's going on in these droplets that lets us track every single transcript that is in the cell, and we also know what cell that transcript comes from, and we also know how many transcripts every individual cell has for each of the genes that it was captured. So the question, and really the magic here, is what's happening in these droplets when we're doing the RT. And as I had mentioned, for indrop, this diagram at the bottom is for indrop specifically. This is actually a slightly older indrop technology. This is V1, I think. There's become V2 and V3. 10X has their own hydrogel beads that are slightly different. The principles across all of these techniques are the same, but the exact primer sequences and all that is obviously going to vary between the technologies that you work with. But the idea is this hydrogel bead in the bottom left corner is embedded with many, many billions of copies of DNA. And every one of these small DNA molecules has kind of two key features on it that I've outlined with these blue arrows. Every one of these beads has a cell barcode where every single oligo on a single bead has the same exact cell barcode. This means that when you do your RT, any cell that's in a droplet with this bead will have the same cell barcode. And that's how you can map each transcript to an individual single cell. The other thing that it has is a UMI. This is a degenerate random sequence. And this is completely random on every bead. So now when you capture a single transcript, each single transcript will get tagged with its own unique molecular identifier. That's what UMI stands for. And now we can take, after PCR, a single transcript might be amplified to 10 or 1,000 copies. You can collapse down all of those copies to a single count based on this UMI. The third more obvious thing, but just as important thing on these beads is oligo DT. And it's this DT, just like if you were doing RT for qPCR, DT is annealing to the transcript's poly A tail and causing the reverse transcription reaction to start, because there's also reverse transcriptase in here. So when you reverse transcribe your mRNA transcript to the cDNA, now you have a single piece of DNA here up the top that has this cell barcode, it has a UMI, and it has the actual transcript that you're after. And it's those three pieces of information that you're just keeping track of when you sequence and process the data in order to build out your gene expression matrix. So the processing that we're not going to handle these or go over these steps today. This would be done through Dolphin Next, and I think I'll pull that up in a second. But the idea is that on the left, very left side, you can see that there's many different colors of cell barcodes here. These are based on which droplet was put with which bead. You have the UMI that tracks the transcript number, essentially. And then you have your cDNA sequence. So these are the three pieces of information that we're keeping track of. Once you align these reads, you can then group them by cell. So here, before the colors were all mixed in, now we can break each color or each cell barcode to its own unit. And for cell 1, we can keep track of exactly which genes we detected in cell 1. And we can do that same process for cell 2, cell 3, on, and build up a digital gene expression matrix for the experiment. So this is what, I'm sorry for the quality here. I guess I blew this one up a little bit too much. But this is what a digital gene expression matrix would look like, or a UMI table, you might hear them called. You have columns, which are cells. So this is all of the single cells that were in your experiment. And you have rows that are genes. And as you can imagine, if we look at gene 2, in cell 1, we have low counts. In cell 2, we have high counts. So this might be a gene that's an indicator that cell 2 is some unique cell type, and cell 1 is a different cell type. And this is the general idea. We won't have this huge matrix that has thousands of cells, tens of thousands of genes, and we want to be able to group together cells that seem to be the same cell type, and then ask questions, okay, what changes in this cell type in association with, say, a disease state or an experimental condition that you might have in your experiment. So how we get to this UMI table from the raw FASTQ files will depend certainly on the technology that you're using. But luckily, a lot of this can be abstracted away from Dolphin Next, which Onur and Alper and Artur, you guys have shown them Dolphin Next, correct?
Yes, we did.
Yes? Okay.
Mhm.
So I'm going to pull up just an example run that I had done before, just so you can see roughly what goes into it. So this is a single cell. Oh, actually, I'm realizing, are you guys seeing Dolphin Next, or are you seeing?
We do, yeah.
Okay, I
Yes.
did my whole screen then. Perfect. So here's an example run. This was all run on the cluster. I'm not going to go into the details of the kind of um, administrator side, which is setting up which environment you're going to do the run in, whether it's on the cluster, on Amazon, etc. I won't go over that as much. But what you can see is that for a given run, you essentially just keep track of a few different parameters. So do I want to use HiSat, or do I want to use star to align this data? Do I want to split the FASTQ in order to parallelize it and make it a little bit quicker? Which genome do I want to align these reads to? All of these are just parameters that are drop-down menu. Just click it, easy enough to do. And it will submit the jobs for you. So here you can see that under the hood, it's submitting all these different jobs to the cluster, to the queuing system, the LSF queuing system in this case, and doing everything that requires you to interface with the cluster and aliquot resources. It'll do all of that for you. All you really need to do is just set up some bare bones parameters to get it to run. So what is Dolphin Next doing as far as each of these individual steps? Usually you're going to start with FASTQ files, which I'm not sure if you guys have seen a FASTQ file, but
Yes,
it
yes,
has.
they did.
Okay, perfect. So you have a header, and then you have your sequence. So we're starting out with BCL to FASTQ, which will take the BCL files off of the Illumina instrument. You tell it what samples you used in your experiment, so what sample barcodes are present, and it will spit out this FASTQ file that we're looking at the bottom. I realized I should go full screen here. The next step in the Dolphin X pipeline that we're going to work with the data from today is extracting the valid reads. So as I had mentioned earlier, each one of those hydrogel beads, it should have a UMI on it, and it should have a cell barcode on it. The cell barcode is actually a whitelist. We know exactly what cell barcodes are in the experiment. So any read that has a cell barcode that doesn't match that dictionary will be thrown away because we don't know what cell that might be. It could be a sequencing error. It could be an error from your polymerase. There are many reasons why you might have some sequence that you just don't know what cell it came from. The other thing it's going to look for is a valid UMI. So on the bead was that 6 mer or 8 mer of degenerate sequences that lets you track each individual transcript. If there's any ends, which the sequencer will do when it's not sure what the base is for that cycle. If there's any ends, obviously we don't know what UMI that is, so we would typically throw that away. So we started with a FASTQ file. We've essentially just filtered and trimmed down that FASTQ file so that now the only FASTQs we have left have a valid cell barcode and they have a valid UMI. Next, you're going to align these reads. Many aligners out there. There's star, there's HiSat, Top Hat, which is older, but we had used for a while. But the idea is that within that FASTQ, we know what the cDNA region of the read is, so which part of the read was for the actual transcripts that we captured. And you can take those transcripts and map them to the reference. So you have a reference, A, B, C here might be 3 exons for gene A, D and E are 2 exons for gene B, and it can just take those fragments and map them to those regions so that you know which genes that DNA sequence corresponds to. And the last step is ESAT. So ESAT is going to take the BAM file, which is just an alignment file from the alignment process. And what it will do is allow you to build your UMI matrix, your digital gene expression matrix. What it's actually doing under the hood is it has reads that were aligned to the reference, and it has an annotation set. So this annotation, you can imagine that if a read aligns to, say, positions 10 to 100 on chromosome 1, that would be an alignment. But you might not know what gene belongs to positions 10 to 1 on chromosome 1. That's what your annotation set is going to tell you. It's going to be a reference that says, for these genome positions, it corresponds to this individual gene. So ESAT will take in your aligned file and some annotation file that tells you what regions of the genome are what genes, and then it will map each of these reads to that reference. And it will build a gene level count. So in the bottom left corner, you can see that we have columns that are samples, rows that are genes. This is the digital gene expression matrix that we're talking about before. And it's ESAT that's taking that BAM file, mapping it to a reference, not actually mapping, it was already aligned, but um, looking at that BAM file and corresponding it to a reference that tells you what genes lie where in order to build up your actual gene expression matrix. So that brings us to the last kind of part of this, is that out of ESAT, you're going to get a UMI table. 10x if you run Cell Ranger, which is 10x's single cell processing pipeline, what you get out of that is a UMI table. So regardless of the pipeline you use to process the data or the technology that you use to generate the data, most of the time, if you're doing single cell, this is going to be your end output, where again, cells are columns and genes are rows. If we think about what a bulk experiment would do to this expression matrix, is that the bulk experiment is just going to be an average of this whole matrix, right? We're not separating out the transcripts from cell 1 from cell 2. They're just going to get homogenized together as one population of transcripts. So what bulk will actually do is it'll be represented by the proportion of each of the cell types that you had in your UMI matrix times that cell type's average expression profile. So in bulk, if say you have 2 samples for a bulk experiment, and in one of the cell samples, there's 50% more of cell type A. If you were to run differential expression, you might say, wow, these genes are all going up. But really what might be happening is just the gene expression is identical, but you just have more of cell type A present in your second sample. So that's something to keep in mind for bulk, that if you try to relate your single cell UMI tables to what bulk would have looked like, bulk is the proportion of each subpopulation in your matrix times that subpopulation's average expression profile. Now, if we were to look at this matrix and kind of eyeball what cell types might be here, you can see that as we've drawn it out there, you might be able to group these cells into 2 different groups, group A and group B. You know, for gene 3, it looks like group B has about 10 times the expression of group A. But obviously, in reality, the matrices are much larger. There's thousands of cells, there's tens of thousands of genes, and you're not going to want to be manually looking at these tables and trying to draw lines in any way. So what we're after is a way to do this kind of in a high throughput manner that doesn't require us to be making judgment calls, and instead allowing the data to tell us what cell types might be present here. And allowing the data to tell us what genes might be important for annotating these cell types. And the other question that we might arise is, now we've grouped ourselves, right? We had group A and group here, B here for each cell type. We might want to store information about the cells in our UMI table or the genes on our UMI table. You might ask, how can we store that information in this UMI table? And the simple answer is that you can't, right? There's no way that we can say cell type 1 is a keratinocyte, because if you look at the gene rows, keratinocyte isn't a gene, right? So we need to just get a slightly more complex data structure in order to work with single cell data. So the package we're going to work with today uses the expression set class. Another extremely common one that you probably will come across if you do any single cell data is the Seurat class, where Seurat is another single cell package that's out there. both Seurat and the expression set class that I'm showing you right now have these exact same data tables in them. They might have a different name, they don't, Seurat doesn't call it feature data. I think it calls it like gene data or something like that. But suffice to say, the Seurat object, while the details of how these things are stored exactly might be slightly different, your Seurat object has these same three tables in it. So just keep that in mind, that the exact data structure we're working with today is a little bit different, but the principles are the same. So what this class has is three key tables. So let's focus on the red one first. Uh, the red one is your assays, or your EXPRS is what it's called in this class, which stands for expression. So this is the digital gene expression matrix, so DGE matrix. It's your UMI table. It's the counts that ESAT gave us, which is each column being a cell and each row being a gene. Now there is also the feature data, which they call F data, is the command to access it. And here, rows are now genes and columns are gene metadata. So if you kind of eyeball these, you can see that your feature data is going to have just as many rows as your expression data, because the rows in the expression data are genes. So your feature data, the rows are genes. So the only difference is that now columns aren't gene expression values. Now they're metadata about those genes. Um, we also have pheno data. So they have it drawn here transposed, but the rows of the pheno data are the cells, whereas in the expression, it was the columns are cells. So here, the pheno data, the number of rows is equal to the number of columns in your expression data. So here, the rows are cells, and columns now become metadata about those cells. So this might seem a little bit abstract, so we can kind of hammer it down and see exactly what does this look like. Here, this data structure, we're going to jump into R just in a minute. So if this, again, seems a little abstract, we'll hammer it down in just a second. But if you run EXPRS command on one of these expression set classes, you can see that genes are showing up as rows and columns are cells. These aren't integer values in this matrix, so this is normalized data. But suffice to say, if a single cell expressed that gene, it has a positive count. Otherwise, the value is going to be 0. When we look at P data, you can see that the column names that I'm showing here become the row names here. So this is just another table. And here you can see that we're keeping track of a bunch of information. I had mentioned we were going to work with bone marrow dendritic cells, where we stimulated them with LPS. So on the far right, you can see a time point column. This is keeping track of, where those single cells were from time point 0 or time point 1 hour. We can cluster the cells and write that in. You can run principal component analysis as PCA, and you can write that information into the P data. So again, anything about a single cell will be stored as a row and column entry in this table. And then last is the F data. So again, rows were genes in the expression matrix, rows are genes here, and we're storing metadata about each one of these genes that we have in the data set. And that's it, actually. So with that, does anyone have any questions about the slides that we just went over?
So I have a question.
Yep.
Could you also clarify the difference between bulk and single cell about the transcript coverage? So, like, when people here decide doing these
Yep.
experiments, how much they can cover with a single cell, how much they can cover with
With
bulk.
bulk.
Yeah.
Yeah, so it's
Mhm.
a bit of a numbers game. So bulk is much more sensitive. Bulk RNA-seq, if you have a cell type above some abundance, it's a little bit of a threshold. But in general, you start with so much material. You have tens of thousands or millions of cells in your test tube before you start your bulk. And so when you extract the RNA and build the library, if a transcript was there, you're probably going to see it. If you contrast that with single cell is, the technology is always improving, it's getting better, but the sensitivity is much less. In that, if you think about our UMI matrix we were looking at, like 95% of the values in this matrix are 0. In a single cell, you might detect 200 to 400 genes, but in reality, a single cell is going to be expressing, at least 1,000 genes, maybe 5,000 different genes. And so you have a sensitivity issue where each individual single cell, you might not see a transcript, but that doesn't mean it wasn't expressed. It might just mean you did not capture that transcript. And that's a good point, Albert, to bring up that there really is a bit of a sensitivity issue. And so it becomes a numbers game. If you have 10,000 single cells, you know, if those cells, say half of them you don't detect the transcript, you're kind of averaging them with the half that you did detect the transcript. So it is something to be mindful of, that bulk is going to be much more sensitive for the genes that are expressed in that sample. Single cell will be less sensitive, but the benefit is you can separate out each component cell type, which you can't do in a bulk sample.
Yeah, yeah, the thing is, like, maybe you can also tell, like, in 10x, for example, in the best chemistry that you can use, can cover only 30% of the transcripts in a single cell.
Yeah,
So
I think
yes,
30 is even a bit high still. I think they're
it,
probably around 10 to 20%.
in their latest chemistry, but yeah, the published data mostly covers 10 to 20%. In drop is 8 to 10%, I guess.
Yep, yep,
So just I want the people understand, like, if you want to work, like, all transcripts in the cell population, then don't do single cell. Or if you're like, if the genes that you're looking for has a low abundance value in a cell, you might not capture that gene. So make sure that what you're doing is actually can captured by single cell. So when you do the single cell, mostly you are getting the transcript, like getting most abundant transcripts in a cell type. So maybe top 1,000 transcripts. So not, you cannot go more than that. So just be aware the limitations of the technologies, and then start working on that. But these technologies are very good to differentiate cell types, or like, to work on the, like, differences between disease versus like healthy cells, that those genes might be highly expressed in the disease, or highly expressed in the healthy cells, but not in disease cells. So usually those abundant genes gives you the story, but not that deep. I just want you to be aware of the limitations.
yep.
Yeah.
And another thing to keep in mind is that if you do decide to go the single cell route, where it really excels is helping discovery. So from single cell, you might get some indication that there's some rare subpopulation. Now you can use that information and run a flow experiment where you sort out that cell type that you've found, and then you can do bulk RNA-seq on that flow sorted experiment. So you don't have to wait about cell heterogeneity. You already know exactly what cell type you're after. So that's a good way where you can use single cell and use it for discovery and finding what niches, populations might be there. But if you really want to hammer down into a sensitive, rare transcript or particular isoform that you think might be involved based on your single cell data, then it does make sense to use other methods. Most single cell papers will start with, single cell on some complex issue, and they're going to use that to drive further experiments. They're going to do mouse experiments. They're going to use flow cytometry or bulk RNA-seq in order to really hammer home that what they think they saw in single cell is actually what's going on. So that's another way where I would not say that they're necessarily mutually exclusive. They certainly both have their use cases. Okay, cool. So
I
now
have
I'm
a
gonna,
quick question too. Can
yeah,
you
sure.
please re-explain the unique molecular identifiers? What is the purpose of that?
Yep.
I didn't understand that. Since we have a cell barcode and we can track each transcript back to its cell, why do we need a unique as well?
Yep. So I'm going to go to this slide. So you started with your cDNA, which was each transcript in the cell was captured by the oligo, and you made a single cDNA. So we then went through these IVT or PCR steps where we're amplifying this library many times. So if we started with 1 nanogram of material, after all of this amplification, let's say we have a microgram of material, we've increased it by 1,000-fold. That means that for every gene, some genes might have been PCR'd more times than others, right? just for example, maybe a particular actin beta transcript was amplified 1,000 times, and maybe some other rare transcript, I don't know, CD14, was only amplified 2 or 3 times, just by chance, because this is all a numbers game. What the UMI is for is that if you started with actin beta 1 transcript, it will have 1 unique UMI on it. So when you amplify that transcript many times, in the end, they'll all be collapsed down to the 1 unique transcript based on that UMI. Does that make sense?
Yeah, so you add the UMIs before the amplification step.
Correct. The UMIs are on there right after the reverse transcription, which happens before any amplification.
Gotcha. Okay.
Yep. So the UMI is used for collapsing down amplification artifacts. Not quite artifacts, but anytime you amplify that transcript, you end up with more than 1 copy, even though it was 1 transcript to start with. The UMI will let you collapse it back down to that 1 transcript. That's also why you will see these matrices that we're looking at called digital gene expression matrices, because it's either a 0 for you didn't see that transcript, or a 1 if you did, or a 2 if you did. So it's a digital gene expression. It's kind of a discrete integer counts for how many times you saw each transcript, and the number of times exactly that you saw it is going to be based on how many unique UMIs you saw for that gene. So here, gene 2, we see 27. It means that there were 27 unique gene 2 transcripts that we saw, and we know it's exactly 27 because of the UMI that's on the bead.
Okay, that makes sense. And another question I had, in regular RNA-seq, there is a fragmentation step. Is there such a thing here, or are the transcripts different lengths?
There is fragmentation, and that is a good point, that when you do this fragmentation for single cell, you need to be a little bit more careful, because on each of these transcripts, on one end, either the 5' or the 3' end, you're going to have your cell barcode, you're going to have your UMI, and you're going to have the nuts and bolts that let you build up the library. When you do fragmentation for single cell, you end up only grabbing one part of that transcript, whether it's the 3' or 5' end. So you have your full length transcript. One end is tagged with metadata. And when you fragment, you're selecting out that part of the fragment that has all the metadata on it. So if we were to look at the, here we go. If we were to look at where the reads map in the data that we're going to work with today, you'll notice that pretty much all the reads map within 1 to 200 bases of the 3' end of the transcript. So this method is a 3' biased method. And the reason is we're using oligo DT to anneal to the poly A tail, which is on the 3' end. And so that's why, just like with bulk, there is a fragmentation, but that's just so that the fragments aren't too long to get on the sequencer. Obviously, you can't have 5 kb fragments on the sequencer. All the clusters will bleed into each other. So you do fragment, but after fragmenting, you're throwing away most of the material, and you're just keeping the 3' end that has the UMI and cell barcode on it. 10x and other techniques have 5' biased libraries, so you're not necessarily tied to the 3' end. But you bring up a good point that ties into Alper's question, which is that in general, these single cell techniques are usually biased to the 3' or 5' end. So if you're looking at particular isoforms in a disease state, single cell might not be the best for that, because you're usually not getting full transcript coverage. There are some methods out there that you do get full transcript coverage, but just be sure that if you're going to plan that type of experiment, that you make sure you can get full transcript coverage if you need it. I saw, I think, one more hand raise.
Yeah. So
Yep.
I have a question. Do you know what is like the average size of these files? Like, is it like, are we talking about gigabytes, like a lot of gigabytes of data for this analysis?
So the files, they're quite large, actually. What I will say is it scales with the number of cells in the experiment. So if you have a single RNA-seq bulk library, each row is a single gene, right? here you have many thousands of cells. So the more thousands of cells you have, the file sizes expand quite a bit. What I can say is that the ones we're working with today, which are only about 3,000 to 4,000 cells, so today that's relatively a small data set, it's still a full flow cell. So we usually do about 3,000 to 4,000 cells per high output flow cell. And a high output flow cell is giving you on the range of, I don't know, 10 to 50 gigabyte fast queues. The BAMs are, 1 to 5 gigabytes each. And UMI table is only, say, 20 megabytes, but keep in mind that's 20 megabyte text file of just cells that are columns and rows that are genes. So you can imagine a 20 megabyte Excel file is, really large. Does that answer your question?
Yeah, thank you.
Yep. So it depends also what step in this pipeline you're in. So you asked, file sizes. The FASTQ files are larger than the BAM files, which are larger than the end UMI tables. So kind of the file size shrinks as you get further into the pipeline. But certainly some of them are, 1 to 100 gigabytes, and that depends on the number of cells and how deeply you sequenced your sample.
Thank you.